Hello and welcome to another exciting lecture for Foundations of Behavioral Neuroscience. In this lecture, we will be going over vision. So we are going to cover a plethora of things here. We're going to start with introduction to sensation and perception, look at how that works for all the senses. Then we'll talk about the stimulus being light. We'll look at the anatomy of the eye. We'll look at some photoreceptors. We'll talk about transduction. Then we'll look at the central and peripheral vision and do the optic nerve, and then a few of the side things or the issues with vision. Before we get into talking about vision itself, we have to talk about sensation and perception. So sensation and perception, which by the way is a course all of its own, you could take a sensation and perception class, and everything we're going to cover over the next few lectures you could cover over an entire semester. So just note that this is a much shortened version of what I could go over when we're talking about sensation and perception. But first, let's look at what is sensation and perception. So the senses, they require both sensation and perception. First, we have sensation. Sensation is where the cells of the nervous system that are specialized to detect stimulus from the environment are essentially stimulated. They are activated. This is where the neurons, or the cells at the tips of the neurons, the sensory cells, are activated. And this works for all of the different senses. And we could look at what the senses are, and we'll go over them more in the next few chapters. But basically, there's more senses than we even normally talk about. So there's going to be things like kinesthetic sense, ability to determine like up from down. When we talk about touch, we're not just talking about touch though, we're talking about touch, temperature, and pain. Sometimes those are conjoined or grouped together into one sense, but they're also often grouped together into separate senses. So these are the different senses we're going to be looking at, and all of these senses have specialized cells that are the ends of essentially the nervous system that activate in the sensation process. The sensation process, though, is just that. It's just the senses activating to the stimuli from the environment. The other side of it, the just as important, is the perception side. And that is, so sensation activates these neurons, the electrical impulses fire into the brain, our brain processes this information and interprets this information from the senses into knowing what is going on in the world around us. Some of this perception is conscious, some of it's subconscious. It's all different types of things that are going on all at the same time. For instance, when we see something, you have the sensation is the electrical, the light hitting the eyes, the electrical impulses in the sensory neurons activating. Then the process, it goes back into the brain, and the brain processes it into perception, into what we see. And it's also important to note, like I said, perception isn't always conscious. There's a lot of things going on with perception that we aren't even acknowledging or knowing that they exist. But our brain is still processing it, even if it isn't part of our focused attention. So this is the system. Sensation is the stimulus hitting the stimulus from the environment hitting the senses, and then it is transferred into the brain and perception occurs from there. Now let's talk about vision itself. So if we're talking about vision, the first thing we have to talk about is the stimulus. So we've got sensation. Part of sensation is the stimulus from the environment. For vision, the stimulus is light. So the photoreceptors in the eye, they will detect visible light, and that is the light hitting the different receptors in the eye. For humans, it's a very narrow band. It's a narrow band of the electromagnetic spectrum that we can see. When you look down here at the bottom, it is this narrow band right here in the black bar. There is a wide range of electromagnetic radiation, humans can only see that very small area. However, we can, with devices to measure all of these. And other animals that have different types of eyes, they have different photoreceptors in their eye, they can see into the ultraviolet and into the infrared. So when we're talking about these, the way the vision works, humans have a very narrow band. There's 3 dimensions to light that are important. Hue is going to be the wavelength. So the wavelength of light, or how close together the waves are. Essentially, a way to say that since we're talking about the electromagnetic spectrum and we're talking about wavelengths, the hue is going to be the wavelength, so the length between the waves. And as you can see from what's here, the higher the wavelength, the more we get towards the reds, and the less we get towards the purples. So it's a wavelength as it goes through the rainbow. Then we've got brightness. Brightness is going to be the intensity of the light, so how strong the wavelength is. And saturation is the purity of light. What does saturation mean? What does purity of light mean? That means how much of it is, of what we're seeing, is one specific light, or is it a merging of multiple different lights? Is it just one specific, or is it a merging? We can't talk about vision and any of the senses actually without talking about transduction. So transduction, the definition of transduction is the process by which biological cells convert one type of signal or stimulus into another. So vision, it comes in as this wavelength. And you've got the wavelength, that's the distance between the waves. You've got the amplitude, that's going to be how strong it is. The saturation is going to be how much of one color or another. But the point here is going to be, though, that transduction is what takes these waves and converts the waves into electrical signals. Transduction transfers the electromagnetic waves of light into electronic signals through the neurons. It transfers the wave into neuron firing. So again, just what's here, this wavelength, color, amplitude is brightness. And transduction takes these waves that we've got here and converts it into electrical signals in the neurons. Let's look at eye anatomy for a little bit. So there's quite a bit going on this slide. We're not going to get too much into everything, but we are going to talk about what's here. So some things to look at. Let's use white, see how white ends up. So no, we can't really use white there. We'll use purple. So you've got the sclera, which is this purple region out here. In this region, I originally wanted to use white because it is a white area. It's an area that is not, that light cannot get through. However, we have in the center here the cornea. And the cornea, in the cornea area, light can get through. And light is then let through the iris, which is the muscle that adjusts the pupil size. So we've got the pupil here. Let's use blue for the pupil here. We've got the pupil here, pupil area here. The iris is the muscle that basically adjusts the pupil size. and this is the what allows light into the eye. The bigger the pupil size, the more light is let in. Then as it comes in, we've got it coming in here. It's going in, it's then focused on the lens. So the lens focuses on the retinal layer, and then once it comes in, it's focused to the various different regions, depending on where the light came in from. So again, the retina is we'll get to the retina in just a second. The iris is the muscle that allows it through the pupil. Then it's focused on the lens, and it goes from there to the layers of the retina, which is in the back here. The layers of the retina contain the photoreceptors that are responsible for transduction. So these photoreceptors here are what take the wavelengths and convert them into electrical signals. One thing we need to talk about whenever we talk about the eye is the optic disc here, which goes to the optic nerve, which is the blind spot back here. So this is an area where there is no photoreceptors, so it's an area where we have a blind spot. If I had more time, I'd talk about blind spot more, but we're not going to go into that too much other than just note that the human eye has a area where there is no photoreceptors. Now we could talk about these other regions here, but like the fact that you've got an area here that basically seals the eye so that things can't get back behind the eye. Even if you get a hair in your eye or you've got a contact lens that gets out of place, it can't move behind the eye because it's sealed there, things like that. And as I said, I could talk all day. I could talk an entire lecture just about the anatomy of the eye. We're not going to do that. We're trying to keep it relatively short. Next, let's look at retinal circuitry, and there's a lot to talk about here. So we talked about those photoreceptors. So the photoreceptors that are in the back of the eye, they are there with, there's also a bipolar layer and a ganglion layer. These other layers, the ganglion layer and the bipolar cell layer, they're translucent. So light passes through these layers into the photoreceptors. And so light is coming in this way, light is passing through the other layers into the photoreceptors. And once it gets to the photoreceptors, in the photoreceptors, there is cones and there is rods. Cones are going to basically be mostly in the center of the eye, the center back of the eye, and they're going to process fine detail and color. So these are the cones are what's going to process color and details. The rods, on the other hand, are much more numerous, but there isn't many, if any, in the center of the eye. They're more around the outside edge. So they're more in the peripheral vision rather than the central vision. Most of the central vision is cones, most of the peripheral vision is rods. There is many, many times more rods than there is cones, and rods are basically responsible for processing the overall picture, and they process black and white. They do not process color. They also work with low light vision, whereas cones do not work with low light vision. There's other types of cells that are going on here. We're not gonna talk about these cells too much, other than just note that when we're looking at some things going on, there's going to be red-green cells, there's going to be blue-yellow cells. Red-green are going to signal red or green, blue-yellow are going to signal blue or yellow indirectly. There's on cells, there's off cells. These determine whether an object is light or dark. There's just a lot going on with this. As I said, I could talk about this all day long. just the different horizontal cells that are in there, the amacrine cells. If you're interested more, the book has a little bit more information. Google has a lot of information. It's one of those where there's a lot more you can get that way. The next thing we'll talk about is fovea, and this is essentially the easiest way to explain it is it is the center of the eye, or the focus of where vision is. It is the area of greatest focus and detail, and it is where basically the ganglion cells process information from fewer ganglion cells, so the information gets processed as a more focused. Instead of, so again, I could talk about this for a while, but the information that comes in, it goes in, it gets to the ganglion cells, it goes to the bipolar cells, and then it goes into the cones and rods. However, when you're dealing with the periphery, the light will come into a ganglion cell, then it'll branch off from there, and then even from there it branches off more, so that one ganglion cell goes to multiple different cones and rods. However, in the center of vision, it is much more focused, and because it's more focused, you get this result of the light will hit one ganglion cell, it'll go to one bipolar cell, and it'll go to one cone or rod, typically again cones here. Allowing it to process more information. Let's look at the rods and cones real quick. So we'll start with the rods. The rods are fairly simple. They're fairly basic actually, because they're basically processing light and dark. So they're processing black and white, light and dark, whether light is present or not. So the big thing that goes on here is what's referred to as rhodopsin. I cannot talk today. Okay, but what the important thing with rhodopsin, yes, now I can say it, rhodopsin, is that it basically activates when light touches it. And that's what you see on the right here. So when light touches it, it activates. Because it's active, it closes the sodium channels. Because the sodium channels close, the ions can't enter, and the results, they become hyperpolarized, and essentially glutamate release is reduced, and it tells the brain that light is present. In the dark, it is inactive. And because of that, the sodium channels are kept open. It allows sodium to flow into the rods, depolarizing them, and they continually release glutamate. So these are the two different states that you have for rods. Either light is touching it, and the rhodopsin, if I can say it today, rhodopsin molecules become activated, or light is not touching it and they are inactive. And that results in the transduction, where the waves are touching it and the electrical signals are sent out based on that, or the lights are not touching it and the electrical signals are not sent. Whereas rods are simple, cones are complex, or at least relatively complex compared to rods. So when we talk about color vision, we're talking about the cones. There are three types of cones that we're going to discuss: red cones, green cones, and blue cones. But also important is the ganglion cells, because they transfer the information and they result in activation. So we've got red-green ganglion cells and blue-yellow ganglion cells. So when we're looking at what's going on with color vision, it's not just one thing that's responsible for color vision. It's not just the cones, it's also the ganglion cells and the cones. So let's look at how these work. So the red light, let's say red light is coming in. So red light, it stimulates the red cones. So red is coming in, we'll deal with red. So it stimulates the red cones. But it also changes the ganglion cells because they can go red or green. That's the thing with these down here. With the ganglion cells, they can go one or the other. They can't be both. So the ganglion cells, it turns them to red. And now that the ganglion cells are signaling red, the red cones are activated, we see red. Let's look at green light now. Green light stimulates the green cones, but as far as the red-green ganglion cells, it turns them green. It switches them to green. So it inhibits red and signals green, whereas red inhibits green and signals red. So by that we see green. How about blue? Blue is easy too. So blue, it stimulates the blue cones here, and it turns blue, the blue ganglion cells, the blue-yellow ganglion cells, to blue. It stimulates the blue, yellow is inhibited, and it signals blue. So that's how we get blue. However, yellow, you see there's no yellow cones. So what happens with yellow is a little bit different. It actually stimulates the red-green equally because it's a mixture of red and green. It stimulates both of them equally. And it essentially doesn't inhibit the red-green ganglion cells. It doesn't excite them. It makes them neutral. So it comes out even. It comes out neutral. Since they cancel each other out, the brain knows that something that is not red and green is going on. But now what ends up happening beyond that is when you look at the blue-yellow, it's signaling yellow, but it's essentially exciting the blue-yellow and it's signaling yellow. So even though no cone is activated, you've got a signal of yellow on the blue-yellow, you've got an even on the red-green. The result is it translates into the brain as yellow. So when we're looking at it, red, green, and blue, they come really easy, but yellow actually has to go through a more complicated process. And essentially what happens is the red cones are activated, the green cones are activated, but the blue cones are not activated, and the brain knows then that it's yellow going on. Another thing to consider is how color vision works. That is, color vision works in such a way, and as I'm talking, stare at the left picture. Stare at the green apple with a red leaf. Just stare at it intently while I'm talking. So the way, and don't look away from it, just stare at the plus sign. So the way this works is that your visual field, the cones are activating, the cones and ganglion cells are activating in such a way. Specifically, the ganglion cells are activating in such a way that you're activating green in one area, red in the other. And then after about a minute of looking at it, staring intently at it, I'll give you a little bit more time, but when you're looking at it, these cells are activating. Then if you look over to the right, maybe even you can look there, you can blink a little bit, look in the white area, you should see a red apple with a green leaf. The reason this occurs is because while you're looking at the left, the ganglion cells are activating in such a way where green is in one and red is in the other. When you look at the white, it basically refracts. It, for a period of time, goes in the opposite direction. And that's why you get this where you were looking at green before, now you see red because the green-red ganglion cells. And where you saw red before, you now see green because, again, of the green-red ganglion cells. So when you're doing this, that's why you see after images quite often of things, because your brain is still, you activated certain ganglion cells to an extent that they basically are bouncing back. They have a rebound effect. So for a period of time afterwards, you'll see the opposite. So again, what went on there is it's the rebound effect, or the rebound firing rate of ganglion cells. While you're looking at the original image, your ganglion cells are firing off, and then they rebound in the opposite direction after you look away. Next, let's talk about the visual pathway. So we've talked about the eyeball, we talked about the retina, we talked about the cones, the rods, what's going on there. Well, then information travels from there down the optic nerve to the LGN, the lateral geniculate nucleus, from there to the primary visual cortex, from there to the visual association cortex, and then it splits into the dorsal and ventral association streams. Let's now analyze each of those. First, we'll start with the LGN. This is part of the thalamus. So if you recall, the thalamus is what sorts certain sensory information and sends it to its proper place. So for this, what is occurring here is that the information that's coming in from the eye, there's two different pathways. So it's drawn there, but let me actually show how this occurs like this. And we've got here as well. So you've got the red pathways going to the right and the blue pathways going to the left. It needs to be noted that each eye sends information to both hemispheres of the brain. Some of it goes, some of the layers go contralaterally, and some of them go ipsilaterally, meaning contralaterally they go to the opposite side of the brain, ipsilaterally they go to the same side of the brain. There's 6 layers of the LGN. 3 layers is responsible for contralateral, 3 is for ipsilateral. And then the pathways that are going through here, you could look at the magnocellular, which is looking at the big picture. This is layers 1 and 2. The parvocellular, this is parvocellular is going to be layers 3, 4, 5, and 6, and this is going to look at fine details in color vision from specifically the red and green cones. And then you've got the canio, I'm just having trouble with words today, sorry. Coniocellular. I don't know why my speech pathways are so difficult today, but the coniocellular pathway is responsible for color vision from the blue cones, and it is located between the 6th main layer of the LGN. So you've really got 3 types of information coming out of this. You've got the big picture information, you've got color information of red and green, and you've got color information of blue. And again, it's both contralateral and ipsilateral. That's important to note. And you also have to recall, we haven't talked about it, but vision is flipped when it hits the retina. So because it's flipped, it's basically upside down as the information's coming in. But our brain processes it as right side up. After it gets through the LGN, it goes to the primary visual cortex. So the primary visual cortex, also known as the striate cortex or the V1. So the striate cortex is the cortex that is basically the one that's going to be responsible for the major visual information. It's responsible for processing the fine details in the major information coming in. After it goes through the primary visual cortex, it then goes to the extrastriate cortex, or the secondary visual cortex, the visual association cortex. We'll talk about that in a minute. But a couple areas that are important in the primary visual cortex is the blobs. Basically it's the cytochrome oxidase blobs. The easiest thing is just to say the blobs. In these blobs, they process color vision. Outside of the blobs, though, it processes the things that aren't related to color vision, like motion, depth, orientation, brightness. And outside of these blobs, you can have simple cells, you can have complex cells, you can have hypercomplex cells, and each of these process various different information in various different ways. Let's look at these. First is the simple cells. Simple cells, the easiest way to describe simple cells without going into this in too much detail is they process line orientation. So the simple cells process whether lines are oriented in a specific way. And there are simple cells for each orientation. So there are simple cells for up and down, left, right, all the different orientations you can have, you have different simple cells for. And if the light coming in is in a specific orientation, so whatever it is that's in the visual field, and you got to think about this for a second. In your visual field right now, you're seeing all kinds of things. You're seeing the writing, and the writing is pretty big. There's all kinds of different lines, but you're seeing lines that are like this. You're seeing lines that are like this. You're seeing lines that are this way. You're seeing all the different orientations of lines, and your brain is processing this based on a whole bunch of simple cells in the primary visual cortex that are activating or not activating. And if it's activating, if they are activating, then there's a strong response to those, and our brain processes that as a line in that direction. There's also baseline when there's no stimulus. So they're actually activating at a baseline level when there's no stimulus. If there is light coming in and it's not at that orientation, it actually shuts off the response. And in a sense, that is telling us as much information as well, because that is telling us that it is not that orientation and our brain is interpreting that. Whereas if it is at the proper orientation, you get a strong response and it's activating that way. So again, simple cells, they tell us the orientation of a cell, of a line. Complex cells combine the information from the simple cells as well as process information about motion and location. So it's taking, okay, you've got a bunch of simple cells that are saying there's a line that's this way, there's a line that's this way, and there's a line that's this way. It takes this information, and maybe this is in motion, so it's moving. This triangle is moving across the screen from left to right. The complex cells are taking that information from the simple cells, mapping it out to a triangle, mapping out its motion, and conveying the information that that is now a triangle that is moving. And finally, you get hypercomplex cells, and these are very highly specialized simple cells that are basically going to tell us the information about the edge of an area. Whereas when we look at, let's say we look at a face, we've got a face, very simple face. We've got simple cells processing information about the mouth, the nose, the eyes, all the different things, but we've also got simple cells that are specifically telling us about these edges, the information at the edges, and even the information at the edges of the eyes too. So these hypercomplex cells are giving us more information about the edges of cells. Okay, after that information has gone through, it goes to the extrastriate cortex or the visual association cortex. That is, it spreads out from the primary to the extrastriate to the out from that primary area. This is often called the V2, and this is where it is our brain is starting to identify the visual information. So in the primary, the visual information is basically coded. It's turned into information. The visual association is where it's taking that information and processing and identifying it. And then it splits it into the dorsal stream and the ventral stream, basically going to the different parts of the brain from there, whether it's going to the parts of the brain that are responsible for processing the information in different ways. That depends on whether it's going to go on the dorsal or ventral. The dorsal stream specifically is the where pathway. So when it's going along the dorsal, or along the top of the brain, that is telling the brain where the information is located. This is also the perception of motion, but it's the where pathway, where the information is located and how it is moving. So the dorsal stream is where we perceive motion. In its spatial location, it's processed and exchanges information basically between different areas. We've got the medial temporal, the MT, the medial superior temporal, the MST. The medial temporal is also known as the V5 since we're talking about the V areas. You don't need to know those exact names. Just note that the dorsal stream is the where pathway, and it also responsible for motion. Conversely, the ventral stream is the what pathway. So the what pathway is identifying visual information, telling us what we're seeing. If the dorsal stream tells us where the information is, the ventral stream tells us what the information is. And an important component of the ventral stream is being able to, this part of the brain needs to be able to identify objects. There are various different parts of this area that are responsible for identifying different types of things. There are certain areas that are identifying objects. There are certain areas that are identifying faces. So we'll talk about it in a minute. Things like the fusiform facial area, and how another thing here is damage to this area can result in visual agnosia. And what that basically means is not being clear. So it makes the vision unclear. And even if the vision is crisp and sharp, you actually, if you've got damage here, you might not be able to identify objects by sight. So this specifically occurs when there's damage to the inferior temporal cortex, which is a part of the visual association cortex, part of the ventral stream, the what pathway. So damage to this, you might be looking at something on the table, something like an apple. We looked at an apple earlier. You look at that apple, you can process this as an apple. Someone with damage to the inferior temporal cortex will be asked, okay, what is that? And they look at that and they can't tell you what it is. They can see it. They can see the features on it. Their mind cannot process what they're seeing. However, if they reach out and touch it, they can tell what it is by touch. If it's something that makes a sound, they can tell what it is by sound. They just cannot, the brain cannot process that identification. So it's a really interesting thing. And that's the visual agnosia. Let's look at a specific part of that. a specific area of this is what's referred to as the fusiform gyrus, or the fusiform facial area, as it's sometimes called. And it's area of the brain that's responsible for being able to determine faces. What can happen with damage to this area is what's called prospopagnosia. So prospopagnosia. It's written out as prosopagnosia, but it's prospopagnosia. I'm even saying it wrong. It's prosopagnosia. I just cannot pronounce anything today. So the prosopagnosia, it's with damage to the fusiform facial area, and what you get here is an inability to identify faces. And just really interesting here, I dated a girl once who had this. She had a traumatic brain injury. She'd been in an accident when she was 17. She was playing a sport and she fell and she hit her head. She had damage to this area and she could not tell faces anymore. This had happened well before I had started dating her, but when I started dating her, she explained this, and she explained how she cannot identify faces at all. She actually goes by features. So she will go by sound, but if she doesn't, if someone's talking, if someone's not talking, she can't identify them. So she'll look at facial features like beards or glasses. Or she got really good at recognizing what clothing people she was around a lot had. So if, like, for instance, at the time I was wearing a lot of very geeky t-shirts that just various different ways that had geeky things on them, and she could recognize me by besides the fact of recognizing me by my goatee that I had at the time, she could recognize me by the shirts that I was wearing, because I wore distinctive shirts. But if I had wore a shirt that I didn't normally wear and shaved my facial hair off and didn't talk, I could walk right up to her and she would not be able to recognize me. Other people who have prosopagnosia, they will do things like look at how people walk and be able to identify how people walk. It's really interesting how the brain can make up for damage here, but it's also very interesting that damage to this small part of the brain means that you just cannot identify faces at all. So let's look at this picture. So someone who has so what you should see here, if you don't have any type of agnosia, what you should see here is a bunch of fruit plus a face. A bunch of fruit making up a face. However, someone who has appreciative visual agnosia, not prosopagnosia, but visual appreciative agnosia, what they will see when they look at this, they'll still see the face because they don't have damage to their fusiform facial area, at least if they don't have damage to their fusiform facial area. They will see a face looking at this, but they won't be able to identify the fruit or the pieces that make up this. However, someone with prosopagnosia is going to be the opposite way. They'll see the fruit, but they won't see the face. So it's just some really interesting things when you're looking at this, how different damage to different parts of the brain can result in different processing. Okay, as I said, I was going to keep this relatively short. I could go on and on and on about vision, but I wanted to keep these topics short, especially since we're behind. So I looked at what is sensation and perception. Started with the stimulus for vision, which is light. Looked at the anatomy of the eye, photoreceptors, the different things that happen with photoreceptors, including the ganglion cells. Looked at transduction, how that occurs. Looked some with central and peripheral vision, the cones and the rods, and how that different things work. Looked at the optic nerve, and all the way going back to some of the issues that can come with vision as we go into the brain deeper. Thanks, come on back.